Seguiram-se, a partir de 1994, as bolsas de investigação BIAL, procurando apoiar a investigação no homem saudável para não haver confusão com os interesses dos laboratórios BIAL. Investigação essa em áreas não muito apoiadas, de grande potencial de desenvolvimento e com interesse para um melhor enquadramento dos seres humanos perante si próprios, perante a humanidade e perante o universo, a psicofisiologia e a parapsicologia.
My name is Mark Blagrove. I'm a professor of psychology at Swansea University in Wales, and I'm also director of the sleep laboratory at Swansea University. We have a 2-bedroom sleep laboratory there. We've just completed a very large project with BIAL, and the results we're presenting today are about daydreams and how they differ from REM sleep dreams. And what we knew about daydreams is daydreams have our everyday concerns in the daydreams, and they also have important events that we're thinking of in the daydreams. We know that. But what isn't known is when in the previous time span those events or concerns from waking life occurred. And so what we did was we had people completing a diary for 10 days, and in the diary they would write down what their concerns were, any important events, what their activities in the day are. And then on the 10th day they came into the sleep lab, they lay down on the bed, and we obtained a daydream from them, and then they fell asleep, and we obtained REM sleep dreams from them and non-REM sleep dreams as well. And what we found for daydreams was that people tended to dream of concerns that had happened to them from the day or the day before coming into the sleep lab, much more than concerns from previous in the week. And what we also found was that people were dreaming of events, people had in their daydreams events that had happened to them. They thought about the events, but the events could have happened any time in the previous week. And so this is very different from dreams. What happens with night dreams is you tend to dream of things that happened the day before, or the day before that possibly. And so daydreams have concerns in them in a way that they don't occur in your dreams. The other thing we did was we looked at the amount of concerns that went into dreams and the amount that went into daydreams, and they went into daydreams much more than into dreams. And it's as if the dream is much more complicated. The dream is a fictional plot that you have to develop, and it's much less reliant on your waking life concerns to create the dream. So the theory behind this, that this supports then, is that daydreams are very different from rapid eye movement sleep dreams. We're very grateful to BIAL Foundation for this because of course it is expensive having people in the sleep lab, and they have to sleep all night. They're woken up repeatedly to ask for their dreams, and they had to keep a diary beforehand. If I could describe the BIAL Foundation in one word, it would be open-minded.
Hello, my name is Helene Wabe, and I'm the Director of Research at the Institute of Noetic Sciences. My BIAL project is called A Physiological Examination of Full Trance Channeling. Channeling has played an important part in human history for a very long time, and you can see examples of it in many different cultures around the world. Some studies have already looked at the truth of channeling, information sources, and physiology of something called a mental channel, but very few studies have actually looked at trance channeling. Trance channels go into a trance state, and they believe that a purported discarnate entity uses their body to communicate directly through their voice or through their body movements. So what my study is evaluating is the neurophysiological measures during a channeling state and non-channeling state, and compares the difference between the two of them. Our participants were connected to EEG, EKG, respiration, temperature, and skin conductance, where they sat in an electromagnetically shielded box. We then went through a lab paradigm where they alternated between channeling states and non-channeling states, with some breaks in between. During those breaks, we asked them, what is your level of consciousness? Are you able to actually witness what's happening? Are you completely in a trance state? And also, what was their perception of how they felt that the disincarnate being was actually using their body? Did they feel like there was full control or just partial control? So at this point, we've actually completed all the channels, which we're really excited about, and we've begun the data analysis. Is there a difference between the channeling state and non-channeling state in the brain waves, the heart waves, and other physiology in the body? We're hoping that this study will shed some light on what's happening in the body and perhaps how channeling works. We are so grateful to the BIAL Foundation, whose support has made this study possible. If I had a word to define the BIAL Foundation, it would be influential. Thank you.
Hi, I'm Aaron. I'm a postdoctoral researcher, and I'm studying the role of dopamine and norepinephrine in behavioral exploration and exploitation. So that was a lot of technical words, and I'll unpack them for you. So exploration and exploitation are how we decide what we're going to do next. So imagine you walk into your favorite bakery, and you need to choose what you want to get. And you could either get the thing that you always get every single time, let's say the chocolate cupcake, or you could try something new that you've never tried before. And that new thing might be better or it might be worse than the thing you always get. So there's a risk involved. And it turns out that there are some chemicals in the brain that modulate how much we choose new things, risky new things, versus consistent old things that we've always tried. Two chemicals in the brain, dopamine and norepinephrine, play a role in this process. And so to get at this, I recorded from the dopamine cells and the norepinephrine cells in the brain, so the cells that release these chemicals. And I actually found that the dopamine cells tend to accumulate activity over when we have many rewarding experiences in the environment. And this accumulation favors choosing the same thing that we always choose. Whereas when I recorded from the norepinephrine cells, which is more commonly known as adrenaline, I found the opposite. So an accumulation of activity in these cells favors trying new things and taking new risks. So if I could summarize the BIAL Foundation in one word, it would be innovative.
I'm John G. Kruth, the Executive Director of the Rhine Research Center, and my study was an exploration of the effects of mood and emotion on a real-world working computer system and network environment. For years, I've been hearing stories about people who are having effects on electronics. People who sometimes can't carry cell phones because they break them. People who have problems with their computers. Some people, even when they walk in the room, computers start working better. I've heard about these things for so long that I started to wonder, is this a real effect, or is it something we're just imagining? Well, this study was designed to test whether this effect was real or not. So the way I tested it is I started out by creating a network. And it was just a computer network that was sending messages back and forth on and on continuously with no one monitoring it, and I was just looking for errors on that network. Also, the second part, I had people come in and do tasks that were designed to induce anxiety. So I had two different processes running. The people didn't know about the network at all, they didn't realize it was running, and that's where I was looking for errors. The tasks they were doing, I didn't really pay attention to because I just wanted to induce anxiety. My two hypotheses were, one, when there are people in the room, I'll see more errors on my network than when they're not in the room. And two, when people are anxious doing these tasks, I'll see more errors on the network than whenever they're not anxious. Because they don't know about the network, all of these effects would be subconscious or unconscious effects on electronics. So if these stories I heard were real, I should see these effects. Well, my first hypothesis, saying when people were in the room I see more errors, it was not significant, not at all, but it was interesting to look at nonetheless. The second hypothesis, that when people are anxious, we'll see more errors on the network, was true. I got a very strong effect. My p-value was less than 0.05, and I had a large effect size as well. It convinced me that if you're working on a computer system and you start to have problems with it, you start to get anxious and frustrated and you start to see more and more errors, it's probably a good time to take a coffee break and walk away. If I had to describe the BIAL Foundation in one word, it would be collaboration.
Hello, my name is Joana Coutinho. I'm a researcher at the Neuropsychophysiology Lab at the School of Psychology, University of Minho, and we just finished this big project looking at the neurobiological correlates of empathy in couples relationships. The main aim of this project was to fill a gap in the literature in which most of social neuroscience studies performed at the moment on looking at the neural basis of empathy use experimental paradigms with fictional targets or imagined targets, or use instead common questionnaires, self-report measures of empathy. Our aim was to go a little bit further in this study and look at the neurobiological basis of interpersonal process as they occur in real interpersonal interactions. So we use the context of intimate relationships, couples relationships, and we had a sample composed by 72 individuals involved in a monogamous relationship with a minimum duration of 1 year, and they performed an interaction task in the lab in which we were measuring their cortisol levels, their heart rate and electrodermal activity while they were performing this discussion about positive and negative aspects of their relationship. Then, and this phase one of the study, looking at the peripheral markers, then we used the video that was record of this interaction to create mini video vignettes that were used for the construction of an fMRI task. So in the second phase of the study, each element of the couple was in the scanner being exposed to video vignettes of the real partner expressing positive and negative communication. So we were measuring the brain activity while they were seeing their real partner, intimate partner. This study allows us to confirm the role of the default mode network, that is a very well-studied brain network at rest for dyadic empathy, and also allow us to see that brain areas more traditionally involved in affective empathy, more emotional brain areas, were more active when the participants were asked to focus on themselves, while brain areas more traditionally linked with cognitive empathy were more active while the participants were asked to be focused on the other experience. So this was a very demanding project that implied a very large data collection with real couples and the construction of fMRI paradigm specifically designed for each participant, which would only be possible with the support of BIAL Foundation, which I really want to thank for the possibility of, even for young researchers, that was my case 4 years ago and still is, but it was a very big opportunity for me, for the development of my career, and also to implement this very ambitious research project. If I had to define BIAL Foundation in a word, that would be going beyond.
My name is Miguel Farias, and I'm the director of the Brain, Belief and Behavior Lab at Coventry University in England. I'm here to tell you briefly about our project, which is called Neuromodulation of Paranormal Beliefs. And the background to this project is really simple. Think about what cognitive psychologists tell us about how we process information about the world. There are two major ways of doing this. One is in an intuitive way, and the other is analytical. An example of intuitive processing information would be falling in love, particularly if it's love at first sight. There's lots of things happening very quickly, and most of the time happening in an unconscious way. On the other hand, an analytical way of processing information would refer to buying a house, for example, in which you have to weigh the pros and cons, how much money you have to buy this place, where can you buy it exactly, that sort of thing. Now, what is interesting about applying this model of dual information processing to beliefs, supernatural and paranormal beliefs in particular, is that in the last 7-8 years, a number of experimental social psychologists and cognitive psychologists have come up with studies suggesting that people who believe in the paranormal or supernatural are much more prone to be intuitive thinkers and to be lower on analytical thinking. They have even done experimental manipulations showing that if you think more analytically in a certain context, you will tend to believe less in the supernatural. So we decided to test this in a causal way by using neuromodulation, transcranial direct current stimulation in particular, which is a harmless, non-invasive method in which you apply electrodes. So it's an electric current that you apply to a particular part of the brain. In our case, we were interested in targeting the right inferior frontal gyrus, which we know it is associated with cognitive inhibition. Again, a process which inhibits your natural impulses or intuition. So we did this with a large sample at our lab in England. And what we saw is that although by using this technique of neuromodulation, it did have an effect in terms of cognitive inhibition, it had no effect whatsoever on the strength of paranormal and supernatural beliefs. We also looked at other measures of intuitive and analytical thinking, and there was no correlation whatsoever with levels of belief in the paranormal. So we did a second study in which we came up with a training program to increase your preference for either intuition or analytical thinking. And again, what we verified that although we could change people's levels of preference, this had no influence whatsoever on people's levels of strength of paranormal belief. So we have now these two studies, and we've published a third study showing that against some of our previous evidence, particularly coming from the US, there seems to be no association whatsoever between dual processing, particularly an intuitive style of thinking, and paranormal and supernatural beliefs. Now, we are very grateful to the BIAL Foundation for having sponsored this project, and if I were to define in a word the charisma of the BIAL Foundation, I would say that it is unique.
My name is Dr. Jason Braithwaite, and this research project was carried out at my previous institution, the University of Birmingham. The title of the project was called The Depersonalized Brain, and we explored psychophysiological correlates of cortical hyperexcitability as related to dissociative experiences such as depersonalization and derealization. The central aim of the project was to explore the role of cortical hyperexcitability underlying disorders of consciousness in non-clinical populations. We did this by exploring those that were predisposed to certain types of anomalous experiences that are commonly associated with conditions like depersonalization and derealization, which ultimately pertain to an unreality of the bodily self or feelings of unreality for one's surroundings. The experiments themselves revolved around a paradigm known as the pattern glare paradigm, which my laboratory has also been developing over recent years. This basically involves presenting striped gratings to individuals, and when the stripes have a certain density or a certain spatial frequency, they can be quite irritating to view. Part of that process can induce visual distortions where you will see things that are not there. The gratings may appear to shimmer, flicker, or you may see coloured halos and distortions. The frequency and rate and range of these distortions is an indicator of increased cortical hyperexcitability and perhaps indicates your predisposition to more hallucinatory phenomena or dissociative experiences. The central findings from this study is that non-clinical individuals who are predisposed to disorders in consciousness and hallucinatory experience do indeed show signs of increased pattern glare. Now, these signs can differ and vary across different dissociative conditions, something which was unexpected in our original hypotheses, but this is an exciting new finding to suggest that maybe going forward we need to fractionate this unitary idea that dissociation and dissociative experience is just one thing. Our research, funded by the BIAL Foundation has shown that there are many different forms of dissociation, and we now perhaps need to start to look at these quite differently. Funding from the Foundation allowed us to hire a part-time research assistant over a 2-year period, which allowed us to gather a wide range of data from large samples to test the role of cortical hyperexcitability in non-clinical populations. If I had to define the Foundation in a word, it would be visionary. It is their vision that has supported our research into disorders of consciousness and how these extend into the non-clinical population, thus expanding our knowledge of consciousness and the relationship between the brain and the mind.
Hi, my name is Julie Beischel, and I'm Director of Research at the Windbridge Institute. Today I wanted to talk about Project 6412, titled Hematological and Psychophysiological Correlates of Anomalous Information Reception in Mediums. The project took place between 2013 and 2015. A little background first. We define a medium as an individual who regularly experiences communication with the deceased. An anomalous information reception, or AIR, takes place when a medium reports accurate and specific information that was acquired without the use of fraud, cueing, or other sensory means. We work with a team of Windbridge certified research mediums who have demonstrated AIR under controlled conditions, and including this study, have participated in 4 BIAL Foundation funded studies. They have an unexpectedly high disease prevalence, including autoimmune disorders and migraines. So that led us to ask the question, and the aim of this study: does mediumship cause disease? And more specifically, do hematological and or physiological changes occur during mediumship readings by Windbridge mediums? This is an exploratory study. It's the first of its kind. The protocol included a repeated measures design with 5 Windbridge mediums participating in 2 randomized conditions, a mediumship reading and a control condition. We looked at 27 hematological factors and basic psychophysiological factors like blood pressure and heart rate. We found that AIR under blinded conditions did indeed happen according to scores given to the readings by sitters. But we found that no detectable hematological or psychophysiological changes occurred during the mediumship readings. So that led us to think perhaps it isn't mediumship that's causing the disease. Mediums also score high on measures of childhood trauma, and a link between trauma and psychic functioning has previously been proposed, and the relationship between trauma and disease is well documented. So it led us to wonder, is it not that mediumship causes disease, but perhaps trauma causes both mediumship and disease? Because of the high cost of clinical laboratory blood tests, this study would not have been possible without the support of the BIAL Foundation. If I had to describe the BIAL Foundation in one word, it would be essential. Thank you.
Our research project is about the Liebert experiment. The Liebert experiment is a very interesting experiment. People are just asked to press a button at a time whenever they wanted to do so. And at the same time, the EEG of them is measured. And we are looking, or what Liebert does is he's looking at the time period before you press the button. So you make a self-initiated voluntary movement, and the question is what happens before. And what you usually see is that up to 2 seconds before, there's a growing signal in the brain that grows until you press the button. So many people thought that's the idea that, and when you, and that's an important fact, we asked the people on the clock, when exactly did you decide to move? And when they then press the button here and they report that they obviously, approximately here they decided. But the interesting fact is that the brain signal started before the decision. So this led many people into the belief that there's no such thing like free will, but that it's like the brain is making the decision by itself. Later on, consciousness kicks in, and then finally you do the movement. So we wanted to know, is this really true? And we replicated this experiment with meditators, with experienced meditators. And why did we use meditators? Because meditators are very experienced in internal observation, introspection, and in attending their own processes. And we wanted to know, how does this decision time and this decision process comes up? And as a control group, we had non-meditators. And we had a meditator who told us how such a decision develops, that there's first an initial kind of urge to do an action, and that you then kind of take up that action and form it into a decision. So we wanted to see whether this makes a difference. We did the experiment 2 times with controls and meditators, and both had 2 versions of the experiment. In the first one, we just told them, press the button whenever you want to press the button. And in the second time, we said, just watch your internal process and press the button then. So in the first version, meditators, the first version is the black line, meditators and controls had the same Libet curve. But in the second time, when we asked the people to pay attention to the internal processes, the curve remained the same for the meditators, but it kind of disappeared for the controls. They couldn't go with this internal urge. And we developed a theory why that is so, but it's a little bit complex, and that will be part of another publication, another grant. Okay? Well, that was for us, this was a very big gain, because we had the possibilities to study this Libet experiment with meditators, and out of that and that experience came many other ideas that kicked a whole new direction in our research group. So we're now doing a lot of research into the field of slow cortical potential and volitional action. And we identified that it's a very important area right now, because there are a lot of new discoveries that link together different fields. And this initial grant gave us the possibility to do this with certain professional attitude.
How would you define in one word BIAL Foundation work and mission?
Great.
Hello, my name is Catherine French, and I'm a researcher at the Champalimaud Centre for the Unknown in Lisbon. My BIAL Foundation project is called Effective Conditional FOXP2 Deletion on Motor Sequence Learning, and the work was carried out between 2013 and 2016. So I'm interested in understanding how our brains enable us to speak and understand language, and one way of doing this is to find out what goes wrong in speech and language disorders. There are some children who grow up in language-rich environments who don't develop normal communication skills. These problems are known to be heritable, but they're normally complex with multiple genes involved. An exception to this is the KE family, where a mutation in a single gene called FOXP2 causes a severe speech and language disorder. This gives us an important entry point into the underlying biology. A core feature of this disorder is something called verbal dyspraxia, which is difficulty making the sequences of movements with the mouth and the face required for fluent speech. We used genetic tools available in mice to remove FOXP2 specifically from defined brain regions. We then investigated the ability of the mice to make sequences of movements by training them to execute series of lever presses. Mice lacking FOXP2 in one brain region, the cerebellum, pressed the lever more slowly, whereas mice lacking FOXP2 in another region, the striatum, executed these sequences of presses more variably. We also found that FOXP2 function during development is important for this skill, because removing FOXP2 protein in adulthood did not affect the lever pressing. Finally, I'm very grateful to the BIAL Foundation for their funding, which has allowed me to realise this project. If I had to describe the BIAL Foundation in one word, it would be progressive.
My research project is the mindful eye and smooth pursuit and saccadic eye movements in meditators and non-meditators. The research was hosted at King's College London in the UK. The main aim of our research was to examine the effects of cultivated as well as dispositional mindfulness on eye movement tasks that are known to index attention and cognitive control. Cultivated mindfulness is used to describe mindfulness developed through training, whereas dispositional mindfulness refers to the naturally occurring tendency to display a non-judgmental present awareness in our everyday life, and it can be measured using self-report questionnaires in the general population. Our findings very clearly showed that cultivated mindfulness, that is mindfulness developed through training, but not dispositional or trait mindfulness, is associated with better attention and sensory motor control. Our findings add to the growing literature on cognitive effects of mindfulness training, and they also suggest that eye movement tasks which have high disk retest reliability and known brain correlates will promise as objective measures of mindfulness training. This research would not have been possible without BIAL Foundation's support for Science Without Borders. If I had to define the BIAL Foundation in one word, it is world-class.
It is a follow-up of a study we carried out, and our main idea is that the ability to become hypnotized and the ability to go into an altered state may be a predictor of when people will do well in a parapsychology experiment. And in this replication, what we found out is that hypnotizability, that is the ability to be hypnotized by itself, was not enough. But if you are hypnotized easily and you go into an altered state, when you are in a situation when you don't have much sensory stimulation, then it makes a difference. So to put it maybe in a sentence, it is not enough to go into hypnosis, you need to go into an altered state and then you do better, significantly better in a parapsychology experiment.
What did this BIAL Foundation grant allow you to do?
What or when? What did it allow? Well, it allowed me to do everything because I have already set up, I have an endowed chair, but what BIAL allowed me to do was to hire a research assistant and to pay participants. So then I have the funds to do this work.
So how would you define in one word BIAL Foundation work and mission?
Fundamental for parapsychology. That was it?
Yes.
That was easy.
It's easy, right?
Muito obrigado.
Thank you so much.
You're very welcome. Okay.
My name is Alejandro Parra. I am President and Parapsychological Researcher at the Institute of Paranormal Psychology in Buenos Aires, Argentina. The title of our research project is Anomalous Paranormal Experience Reported by Nurses Themselves and in Relation with Their Patients in Hospital Examining Psychological, Personality and Phenomenological Variables. We start in 2014 up to 2016. The aim of this study was to determine the degree of occurrence of certain unusual perceptual experience in hospital settings, so-called anomalous paranormal experience, often related by nurses and carers. We hypothesized that nurses who report this kind of experience will tend to score higher on work stress, higher on schizotypic proneness, higher on absorption, and higher on empathy than those who do not report such experience. 344 nurses were recruited from 36 hospitals and health centers in Buenos Aires who were grouped as experiencers and non-experiencers. The most common experiences are the sense of presence and or apparitions, hearing noises, voices or dialogues, crying or complaining, intuition and extrasensory experience, and as a listener of experiences of their patients, such as near-death experience, religious interventions, and many anomalous experience in relation with children. This kind of experience also tended higher on absorption, which was confirmed, proneness to schizotypy, which confirmed, and I have to define the BIAL Foundation in a word. It was enjoyable and relevant in many ways. Firstly, BIAL contributed with us since 18 years up to date, and secondly, BIAL Foundation was the first and unique supporter on parapsychological research of excellence. Thank you.
My name is Lenia Amaral. I'm a member of the research team responsible for the project Episodic Memory Enhancement in Aging, the role of cognitive training combined with TDCS, led by Professor Mário Simões from University of Coimbra. In this project, we were interested in understanding whether and how cognitive training combined with the TDCS facilitates episodic verbal memory in older adults. To do this, we tested if cognitive training and TDCS over left dorsolateral prefrontal cortex or over right cerebellum facilitates memory compared to sham stimulation or a controlled group. Our results show that only the groups receiving both interventions, so the groups receiving both TDCS and cognitive training, shows a significant improvement in the tasks related to episodic verbal memory. Furthermore, the group that received stimulation over the right cerebellum was the group that showed greater improvement and consistency in terms of results. Our data suggests that, in fact, cognitive training and TDCS can be a promising combination to enhance cognitive functions in the healthy elderly. Moreover, our data also suggests that cerebellum could be very important to cognitive processes. This work could not be possible without the support of BIAL Foundation, and if I have to define the BIAL Foundation in one word, it is progress.